All right, everyone. I'm not going to be doing an entire voice recording for this section. The reason being that the slides here and what I would be talking about in lecture follow really closely along with the material in your book. So you should be reading that material from chapter 5 and then kind of going along with the slides to be able to know exactly what you should study and exactly what you should focus on for the next test. So the part of this section that I am going to talk about in a lecture recording here are the last few slides. So this is material that isn't really mentioned too much in your book, but it's still important. These are examples of evolutionary change in human populations in recent times, like over the past 10,000 or so years of human history since humans began agricultural practices. So the first thing that we'll talk about here in this adaptations to a changing diet part is going to be lactase persistence versus something called lactose intolerance. So lactase persistence is the ability to produce the enzyme lactase past the age of weaning. So as mammals, we have the ability, all mammals have the ability to produce this enzyme called lactase in infancy. And basically what happens in all other mammals is that the production of this enzyme lactase ceases at the age of weaning. So whenever that is, whatever, it's going to vary from animal to animal. In human populations, the age of weaning from studies of hunter-gatherers is somewhere around age 2, maybe, when most children are fully weaned. So lactase persistence basically means that an individual keeps producing this enzyme lactase into adulthood. Now, the reason why this enzyme called lactase is important is because it is required by your digestive system to properly digest lactose. So lactose is a type of sugar that's found in milk products. So milk from any species, whether it's human breast milk or cow milk or goat milk, what have you, it all has lactose. And in order to digest that sugar called lactose, we need lactase, which is the enzyme. So basically, you may or may not be lactose tolerant yourself, but if you do possess the enzyme, you can eat dairy products, cheeses, ice creams, whatever, as an adult with no problem. If you do not produce the enzyme, if you're quote-unquote lactose intolerant, what that means is that if you consume dairy products as an adult past weaning age, so even, in childhood, age 7, 8, 9, you could still have a bad reaction. Basically, you'll have severe gastrointestinal distress in the form of nausea, diarrhea, and gas. So the ability to digest lactose in adulthood, so this trait called lactase persistence, means, so the ability to produce lactase as an adult varies from population to population. So some people on the planet have the ability to consume dairy as an adult, but a lot don't. And actually, if we look globally, the highest proportion of humans are actually lactose intolerant. So having lactase persistence is something that's actually fairly rare in the scheme of things. If we're looking at all 8 billion people on the planet, way more of them are lactose intolerant than tolerant. One word really quickly about the term lactose intolerance. The use of the term lactose intolerance is kind of an example of something called bioethnocentrism. Bioethnocentrism is essentially the tendency to consider our own traits as normal for species characteristics. So if you are lactose tolerant, you might consider yourself to be the normal one. If you have a friend that is lactose intolerant, you might consider them to be kind of odd or weird or unusual in some way. So bioethnocentrism is kind of adding a value judgment to physical characteristics that differ from population to population. Now, the reason why the terminology lactose intolerance is an example of bioethnocentrism is because up through the 1960s, medical understanding in the West was that lactase persistence was the norm. So the norm, they thought in the 1960s, was to be able to consume dairy as an adult. Even though doctors at the time, scientists at the time knew that certain populations couldn't, they thought that those were kind of the outliers, those people that can't digest lactase as adults or lactose as adults were just kind of odd and something was wrong with them. That was really that was kind of a skewed version of reality, because we do have heavy frequencies or heavy or like high frequencies of populations in Europe that do have lactose tolerance and do have lactase persistence. Those were the people who were in the medical community, people of European ancestry. So basically they had kind of a biased idea of what was normal for humans. In any case, we now know that most people on the planet are in fact lactase impersistent, meaning they cannot tolerate digesting lactose as an adult. So looking at this map here, the map on the slide is a map of lactase persistence versus impersistence across the globe for the Old World. So what we see here are some pretty distinct patterns. We'll just talk about what the color coding means. The red areas are areas with the highest frequency of lactase persistence, meaning populations where you have a lot of individuals who can digest dairy as adults. So if you can look at this map and see, we have a heavy concentration in Europe, especially Northern Europe. We have a concentration in West Africa here, and then over here in part of the Middle East and modern-day Saudi Arabia, and in parts of South Asia as well. So what we see here is a very non-random distribution. So we have these hot spots where we have lactase persistence, and then we have a lot of other areas as we fade through this color scheme down to blue, where we have fewer and fewer members of the population that are able to digest lactose as adults. So the individuals down here in these areas with the dark blue, those basically less than 10% of the population in those regions can digest lactose as adults. And then, you can see how we can go through the frequencies here up to 90% for the red areas. So again, what we see here is a highly non-random patterning here. Now, from an evolutionary standpoint, it's kind of interesting if we look at the map that we see here and try to figure out exactly why, from an evolutionary standpoint, we would see patterns like this. So basically, the thought process is that the ability to digest lactose into adulthood would have been really useful in certain populations in the past, only if they had a history of animal husbandry. So domestication of animal species that are providing a consistent source of milk. So what we see here is actually the case. We see these areas of the globe here that have a long history of pastoralist behavior. So herd animals, using domestic livestock and practicing animal husbandry. So we do know that hunter-gatherer populations before the rise of agriculture and before the rise of domesticated animals were overwhelmingly lactose intolerant, and all modern hunter-gatherer groups are overwhelmingly lactose intolerant as well. So genetic studies, geneticists have looked at this, and they have figured out basically that these mutations really started increasing in human populations just within the last 10,000 years. So again, we're seeing this genetic change that is in lockstep with the cultural practices that are going on in these parts of the world during this time. So looking here at this map, as you can see, as we mentioned before, we have several centers of lactase persistence. We have one in West Africa, we have one in the Middle East and South Asia here, and then one in Europe. So scientists have wondered what exactly, from an evolutionary standpoint, could have led to this type of pattern happening. What were the selective pressures that were leading to these certain population pockets developing this practice, this cultural practice, and then having the kind of genetic frequency increase as a result. First off, let's talk a little bit about some of the selective advantages to being able to digest lactose as an adult. In Europe, basically, one of the possible selective advantages would have been a strong selective advantage for vitamin D consumption. So this is something that you should be a little bit familiar with if you followed along with the readings and you did the assignment for the week. The assignment was talking about skin color and how it's adaptive in different parts of the world. Basically, we synthesize vitamin D from exposure to the sun. So populations that live further and further to the north are being exposed to lower and lower levels of solar radiation. As a result of that, vitamin D synthesis decreases as you go further to the north. So going back to the concept of milk consumption being selectively advantageous, if you are able to consume dairy products, if you have herd animals, if you have domesticated cattle, for example, they're producing milk. Milk is naturally high in vitamin D. So basically, if you are in a part of the world where you're not able to fully synthesize enough vitamin D from the sun because you're too far to the north to have enough UV radiation, having extra vitamin D in your diet in the form of milk would help keep strong bones and all that kind of stuff. So that could possibly be a strong selective advantage in Europe, which would be why we see this population up here in Europe with about 90% frequency in lactase persistence. Now, if we look at these populations closer to the equator, that doesn't really work. That explanation doesn't really work. So it really wouldn't be a selective advantage to have more vitamin D in the diet in these regions because these people are already getting plenty of sun exposure. They're so close to the equator, there's no selective pressure for them to have to be able to consume more vitamin D. One thing that might be happening in these regions closer to the equator is that it could have to do with water content. So if you're living in an arid desert, as you see here, these parts of Africa and the Middle East are quite arid, it would be helpful if you could consume basically anything with water content in your environment. Milk could be a really good primary source of water at certain times of the year. So basically what we see here is we're probably seeing different selective pressures acting in different places. Now, from a genetic standpoint, multiple studies have looked at the genetics of lactase persistence. It is a Mendelian trait. We talked about Mendelian traits last week rather. It is lactase persistent people have basically the dominant form of the gene. So lactase persistence is dominant. Lactase impersistence is a recessive gene. So all in all, what we see here is pretty strong selective pressure in various places worldwide to encourage the spread and the increased frequency of this gene. The next thing we'll talk about just kind of briefly is starchy diets. So diet changes really significantly with the Neolithic Revolution as we move from hunter-gatherer lifestyles to more settled lifestyles where we have farming and that kind of stuff, we automatically see an increase in starch consumption. So it happens in different places at different times, but as early as 7,000 years ago in China, we have full rice domestication and rice accounting for a large percentage of the diet. We see other patterns in other parts of the world that are pretty similar to that with local domesticates. So in the East, we see rice. In North and South America, maize production, meaning corn. In the Middle East, we see wheat and barley. So there are various things that are being domesticated, but generally across the board, we're seeing an increase in starchy diets. So starch is comprised of a polysaccharide called amylose. Amylose, this term here, is a sugar basically that's being digested in your, is being digested in your digestive system by an enzyme called amylase. So you have to be producing amylase basically to be able to digest, to efficiently digest amylose. Okay, so a diet rich in starch would have been pretty advantageous from a calorie standpoint. So starch is a pretty high calorie food. Back in these times, the more calories, the better. So being able to efficiently digest starch would have been a strong selective pressure. This gene, AMY1, listed here, is a gene that we know to be related to amylase production. So this is a gene that is called, it's referred to as being polymorphic in copy number. What this means is that individuals vary in terms of how many copies of this gene that they have. So some people have 2 copies of the gene, and other people have up to 15 copies of the gene. So there's a lot of variation in terms of how many individual copies of the gene a person has in their DNA. Now, what research has shown is that individuals with more copies of the gene produce more amylase and are therefore better able to digest starch. So what the geneticists have figured out is that populations that have traditionally high starch diets, so those in Japan and China, parts of the Middle East and parts of North and South America, those populations tend to have the highest copy numbers. So Japanese and Chinese, for example, tend to have around 15 copies of the gene because those populations have long histories of starch consumption. Hunter-gatherers, for example, only have 2 to 3 copies because they don't have a history, they don't have an evolutionary history of having to digest a lot of starch. The last one that we'll talk about is alcohol consumption. Alcohol is something that is not only consumed heavily by human populations, but it's consumed by a lot of other animals as well. It's very common to have chimpanzees, for example, or monkeys, various species of monkeys, seeking out fermenting fruit in the jungle, trying to get that alcoholic content in the fermented fruit. Now, we'll go through the bullet points here. The first one, the term alcohol tolerance refers to any of the body's responses to the functional effects of ethanol. So when we're talking about ethanol, we're talking about what actually makes somebody intoxicated. What is the substance that makes somebody intoxicated in an alcoholic beverage? That's ethanol. Now, alcohol tolerance includes direct tolerance, which is basically how much of the alcohol do you have to have before you begin to feel the effects. So do you have 1 beer? Do you have 4 beers? How much does it take to make you start to feel the effects? Alcohol tolerance also refers to the speed of recovery from insobriety. So if a person becomes drunk, how long does it take them to become sober again? And finally, resistance to the development of alcoholism. So some people have genetic predispositions to make them more prone to becoming dependent on alcohol. So those people have to be a little bit more careful than other individuals who maybe don't have that gene. There's also something referred to as consumption-induced intolerance. Basically what this means is that you have a tolerance to alcohol that's increased by regular drinking. So the more alcohol you consume in your day-to-day life, the more resistant to those effects you are. So someone who never drinks, maybe they have 1 drink and they start to feel the effects, they start to feel tipsy. But someone who drinks a little bit every day has a higher tolerance. Okay, so moving on to alcohol digestion. So as it says here, alcohol digestion involves two steps. We have the original substance that you're taking in when you're drinking alcohol, and that is called ethanol. The first step in alcohol digestion involves the breakdown of ethanol into a substance called acetaldehyde. Now, a group of enzymes are responsible for this process. Those enzymes are called the alcohol dehydrogenases, listed here. So this two-step process is being driven by these alcohol dehydrogenases. So we have the first step where we have ethanol being broken down into acetaldehyde. Acetaldehyde is actually a fairly toxic substance to humans. This is a substance that chemically is pretty similar to formaldehyde. Formaldehyde is what they use to preserve dead bodies. So it's a pretty toxic substance. So that toxic substance has to be broken down into something that is harmless to humans. So acetaldehyde is broken down into acetate, which is basically just vinegar. Okay, so we have this two-step process. In most people, it happens at a steady rate. You have this two-step process happening pretty quickly. So you have the ethanol being broken down into something pretty harmless in like a short period of time. Now, as I said before, this two-step process is driven by the alcohol dehydrogenases. The production of these alcohol dehydrogenases are controlled by a gene called ADH1. The ADH1 gene has two variants, as it says here, the A form and the B form. So basically what happens if someone has the first form of the gene, the A form of the gene, is that this process happens with no issues. We have the ethanol being broken down into acetaldehyde, and the acetaldehyde is immediately broken down into acetate. So even though this toxic substance, the acetaldehyde, is being produced, it's very quickly being broken down to something else. People who have the second variant, the ADH1B gene, basically there is an interruption in this process. So the ethanol is broken down as soon as the person consumes an alcoholic beverage. The ethanol is broken down into acetaldehyde, but the second step is delayed. So the second step is delayed to the extent that the acetaldehyde starts to build up in the person's system. As I said, acetaldehyde is a toxic substance, so you end up with this buildup of a toxic substance in the person's body. Eventually, the second step will catch up with the first step, and you will have that harmful substance broken down into acetate, but it's delayed. The second process is delayed in these people with the second variant of the gene. So basically, they tend to get sick. They tend to become drunk quickly. They tend to have ill effects. They get sicker faster. They have something called an alcohol flush reaction, which means that their face turns bright red. Basically, it's a reaction in the body of a flushed face and neck, which is a result of having this toxic substance building up in the body. So in terms of these two variants, we see a non-random distribution of these two variants as you can see on the map here. So the green regions of the map, those individuals in that part of the world tend to have very high rates of the second variant of the gene, meaning they have this delayed process where the acetaldehyde builds up in their bodies. So the way that it turns out is that almost all Japanese, Chinese, and Korean people have that second variant of the gene, and it results in slower metabolism for the second process. So they have, again, this buildup of acetaldehyde in their bodies. Now, from an evolutionary standpoint, when we see these non-random distributions, these non-random patterns on the map, we have to wonder why. So what evolutionary processes are possibly at play here that are leading to this, this distribution? So a study was conducted to try to figure out which is what's called the ancestral trait. So what is our start point? What did humans have 15,000 years ago? And scientists have found that 15,000 years ago, pretty much all humans had the first form of the gene. So having this two-step process happen with no problems whatsoever. At some point between 6 and 12,000 years ago, we see indications of a rise of the second variant, basically. One study tried to kind of tie these two patterns together, and they figured out that this allele, the second variant, basically started to appear and spread along with rice cultivation in the region. So between 6 and 12,000 years ago is when rice was being cultivated and becoming widely consumed. So basically what they thought probably is happening is that as people in this region are cultivating rice, they're also beginning to ferment rice into ethanol. The results of having this increased alcohol availability could potentially have led to a lot of alcoholism and alcohol abuse by those who were able to acquire it, which in theory would result in lower reproductive fitness. So the individuals who were consuming a lot of this alcohol were experiencing lower reproductive fitness. So the idea is that there was selective pressure for individuals in this population to have the second variant so that they wouldn't be able to consume a lot of alcohol. Because again, the second variant means that you get very sick very quickly when you consume alcohol, and it's not really a pleasurable experience. So that is where we'll leave off for today. Please make sure that you're following along in the textbook. As I said, most of the material from these slides is explained in the textbook. So feel free to contact me if you have any questions with definitions or anything. But I double checked before doing this voice recording, and pretty much everything that's in the slide is in the textbook. So make sure that you're following along with the readings in addition to listening to these voice recordings.